We're going to move on right to our next speaker, who is Dr. Marinka Zitnik. Pleasure to introduce her, assistant professor at Harvard University, with appointments in the departments of Biomedical Informatics, the Broad Institutes of MIT and Harvard Data Science. Her research won multiple awards. I won't list them all. The International Society for Computational Biology, International Conference on Machine Learning, Amazon Faculty Research Award, so on and so forth. These, by the way, bios are available online. It's really exciting to hear Dr. Marinka talk to us about how her recent research will create new avenues for accelerating the development of therapeutics, fusing biomedical knowledge and patient data, and giving the right patient the right treatment at the right time to have medical effects that are consistent from person to person and with the results with a laboratory. So we really look forward to this research presentation, how artificial intelligence tool will guide biomedical discovery and produce interpretable outputs that lends themselves to actionable hypotheses. Without further ado, Marinka, the floor is yours.
Hello, everyone. I'm really delighted to be here with you today, and thank you so much for this kind introduction and inviting me to join this esteemed community of speakers and also engaged, enthusiastic attendees. So I'm sharing my screen. I hope you can see it well. Let me know if not. So today I will talk about some of the recent advances in development of core AI and machine learning methodology and how they can really help us advance biomedical discovery and also medical, in a way, have impact in medical practice. Most of the projects that I will be mentioning, including have their associated project websites on our lab website. If you go to zitniklab.hms.harvard.edu, you will find many pointers to data sets, examples, tutorials, code bases. So those who are excited about just trying out some of these algorithms or using them for your own research projects, you're more than welcome to do so. And we provide resources for that through this website. With that, let me begin. So science crucially depends on existence of scientific instruments. And so while in the past, those scientific instruments were primarily physical devices, such as microscopes, that have facilitated discoveries, we are now in the era of modern data-intensive sciences that require new kind of instruments that in many ways I envision will be optimized for knowledge discovery from data. And so we are now in the area where these tools or new instruments are actually not physical devices anymore, but are algorithms or virtual devices that can help us discern patterns that are indicative and meaningful biologically or medically from large data sets that can then be used for prediction. There are many opportunities for AI in medicine and health. Earlier speakers have outlined so nicely many of them. What is important to mention here is that the AI algorithms in particular create opportunities to accelerate and improve efficiency of a variety of different workflows and procedures that exist in the medical enterprise and in the biomedical discovery space. That ranges from preliminary diagnosis, clinical trial participation, improving administrative workflows and costly back office problems that are actually major impediment in health systems, all the way to designing assistants that can help in automated image diagnosis or modeling of clinical notes, modeling of comorbidities and chronic disease treatments, and more broadly, thinking about algorithms that can make sure that the data are used in a robust and ethical manner by protecting the data and avoiding errors. So with all that in mind, you might be wondering, well, are there certain aspects of biomedical data that make biomedical applications or biomedical AI unique, different from many other areas where we see tremendous progress that AI has brought and AI solutions have created. So let me answer this question over the next few slides. There are many aspects that make biomedical data unique, and we need to be very careful when we are implementing and designing AI algorithms for biomedical data. Let me go with you to a couple of them. The first important aspect is that for more or less any biological system or medical problem that we are interested in solving, we typically need to integrate heterogeneous confounded data that can span all the way from molecules and molecular interactions and to the level of societies and interactions within an ecosystem or a population of patients. And so that is challenging because prevailing algorithms that were designed for other non-medical domains in many cases typically assume that the input data will look like one large flat data set. That is simply not the case. We often need to integrate the information all the way from genes to behavior in order to get a good understanding into a complex disease or a complex phenotype. Second, the models that we are developing need to be able to generate predictions that are not only accurate, but that also can be interpreted meaningfully, meaning that predictions need to lend themselves to hypotheses that are actionable, such that users of those AI predictions, whether those are biomedical experts, clinical professionals, virologists, experimental biologists, are able to perform certain actions on them, are able to perform experimental screens in biological lab or redesign or clinical trial based on the outputs that an AI algorithm would provide. So accuracy is no longer enough. There are other important aspects that we need to take into account, such as explainability and also being able to provide predictions that are fair and non-discriminatory. And why is this so important? This is so important because unlike many of AI applications outside of biology and medicine, very often when we are designing AI systems in this area, these systems can be used in high-stakes decision making settings where decisions are made about life and death. And though because of that, these algorithms need to be robust, in many cases provably robust. We need to ensure that models behave responsibly, they're trustworthy, and we need to integrate many checks and balances into deployment of those algorithms in clinical or biomedical implementations. So that is really important and does not arise in all other domains where we also see the use of AI. It is, for example, also important in settings such as autonomous driving. And so the final, the way to kind of look at these unique challenges that biology and medicine present themselves then creates the need for having algorithms that are specifically designed for biomedical problems. And kind of those algorithms can, essentially the way they would operate, they operate on these very large interconnected data sets that can bring together data from very different domains. So here I'm showing on a slide an example of that, where we have the large complex biological system modeled at different scales, from the levels of interactions between proteins at a molecular level, the way those proteins are targeted by small molecules, small compounds that are drugs that a patient is taking, all the way to the level of an individual or a patient that might have multiple coexisting diseases at the same time that they need to be considered into modeling, and further away in the social or environmental aspect that tells us about the lifestyle and behavior of that individual. So that creates this kind of rich, very interconnected biological systems that we then need to learn and reason over them. So this is where AI comes into play. And the question becomes, how can we take these large interconnected biomedical systems as input and then design algorithms that will produce predictions, patterns, insights? So modern AI is based on is really fueled by advances in algorithms that broadly are known as representational learning algorithms or sometimes deep learning algorithms that effectively take these input systems, input interconnected data sets and distill them into condensed summaries or representations that then can be leverage for downstream prediction. And those representations are essentially machine readable, machine computable summaries of the data and biological systems that are understandable to algorithms and to machines that then can combine those representations to derive useful predictions that are accurate and interpretable. I will highlight examples of those throughout the talk. So in my research group, and we are very excited about creating foundational AI models that can be trained on very broad data at scale, and that can act as AI systems that users, scientists, and healthcare professionals can interact with. They can ask questions about whether a particular drug will cause a particular side effect, or will a compound or a drug target a gene that participates in certain pathway. Those different kinds of scientific and medical questions can be asked and to our AI system. What our AI system does is it provides answers to those queries. And it does so by operating on very large interconnected networks or graphs, which we refer to them as knowledge graphs. And then these representational learning algorithms that I will describe in the talk are able to extract useful patterns from these knowledge graphs and provide predictions and explanations for those predictions to domain experts. And so that is exciting because it creates this collaborative environment between the AI system and the domain experts, so the domain expert can interact with the AI system and ask different kinds of questions, and those questions are then being answered by AI algorithms by parsing very large knowledge graphs that might have billions of relationships that are derived from direct experimental readouts and prior medical knowledge. The kind of algorithms that are fueling these AI systems, as I mentioned, are representational learning algorithms. So a class of representational learning algorithms that has really been prominently successful in core machine learning fields in the last 2 or 3 years has been known as graph neural networks. So let me tell you what graph neural networks do through a single slide. So imagine having on the left is our input data, which is given as a set of nodes, set of entities that might codependent interact with each other. Think of those entities might be, for example, patients, and they would be connected based on their physical, familiar, or friendship relationships, or nodes might represent different genes connected to each other based on how they interact. So we have these input data sets on the left. And so a graph neural network is then an algorithm that is described by a nonlinear deep transformation function, here denoted F, that will take each and every component of the input and map that component, which we call node, to a low-dimensional, compact, condensed summary, or a vector of real numbers that has a name, and that name is embedding. So the idea is really, how can we do that effectively? And so what graph neural networks on mapping function F do is they project this input data in this compact embedding space, and they optimize this embedding space such that performing linear algebra operations on condensed embedding vector summaries reflects topology of the input data set. And so that is the bread of the butter, and the butter of how this graph neural network architectures and transformation functions are designed such that two nodes, two components in the input data that are similar to each other, that have similar local interaction patterns, that have kind of similar signatures in our input data, they will be mapped to points in the embedding space that will be close to each other and will have similar embeddings. So that those embeddings then are then machine-readable summaries that can tell the algorithms about what kind of prediction should be made for those objects. So these graph neural networks, or denoted F here, serve that function. They map nodes to those d-dimensional embeddings such that similar network neighborhoods in the input data will be mapped to points in the embedding space that the model optimizes for that are close together. So there are many ways how to achieve that, and our group has been developing a variety of different graph neural network architectures that are based on a concept known as message passing, and sort of this is now a very vibrant, active research field in machine learning more broadly. Broader implications of these advances in machine learning is that with graph neural networks, we can now apply deep learning much more broadly than what was possible before. Sort of since several decades already, we have seen, and now more and more, the use of deep learning for analyzing imaging data, such as medical images, and sequential data, such as biological sequences or natural language. What now we can do is we can apply deep learning to any interconnected data set. And so that creates some fundamentally novel opportunities in biology and medicine that go beyond merely looking at medical images or natural language and biological sequences. So in the rest of the talk, I will show you a few examples and give a couple of vignettes of what kind of tools and applications these algorithms have enabled. So one particular question that we are very interested in is, can we use AI algorithms to improve efficiency of drug discovery and development? Drug discovery and development is an incredibly costly, resource-intensive process. It takes on average between 11 and 16 years, and it costs $2 to $3 billion to design a new drug from scratch. So how can we make this process more efficient, faster, and cheaper? So to this end, what we have designed are very large precision therapeutics knowledge graphs that, as I said, bring together heterogeneous complex biological data sets, and then design graph neural architectures that can, for a given disease, identify what are most likely compounds that are likely to have medicinal effects and would be safe and effective for patients if they would take them. And so particularly exciting it was to see how AI algorithms can perform well and have strong performance for those diseases that have very few treatment options and very limited molecular understanding. Because for those diseases, the need for safe and effective therapeutics is most prominent. And so AI algorithms can help achieve that. On the right top, I'm showing a plot that shows performance of the algorithm measured as AUPRC. This is area under precision recall curve. And on the x-axis, we see different settings. And so I want to point your attention to the second and third setting that show performance of these models in orange, in environments where we want to identify promising therapeutic opportunities for diseases with very few treatments. And we see over 49% improvement in those cases in terms of improving efficiency and success of identifying therapeutic opportunities that down the line end up being more effective for patients than alternatives would be if the algorithm would, if the experimentalist would not be considering AI predictions. So this is really exciting. Of course, we don't want to only have accurate predictions, but we need to be able to provide mechanisms, understanding about how would a particular compound actually work, how would it exert its therapeutic effect on the disease, how it would treat patients. Only if that is if we can explain that mechanism of treatment to users, and it is which would be biologists and chemists and clinical trial designers, it is likely to expect that kind of information can percolate down the drug discovery pipeline and ultimately result in an approved drug. So the question is, well, how can we then summarize where in these large knowledge graphs that serve as the data to train models, the models look for evidence for their predictions? And can we find small subgraphs that are most influential for a particular prediction that the model made, and then present that subgraph as an explanation that justifies why the model predicted a particular compound for experimental testing. And so the way to bring all that together is to design various interactive visual tools. I'm showing an example here on this slide, Drug Explorer, that enables AI scientist collaboration, where domain scientists can interact with AI predictions and their explanations and use them in their daily work to inform the design of experiments and to make decisions in these high-stakes settings. We recently did a user-centric study with our collaborators from Mount Sinai Hospital System in New York City, where we were where we wanted to understand the utility of Drug Explorer in these AI scientist collaboration systems. And the way we did that is by asking users of AI models whether they find explanations and predictions made by algorithms really useful for making decisions about what drug might possibly be safe and efficacious for a particular disease. And so what the results of that were that we found a substantial increase in the trust and confidence of users into AI predictions, where they were shown explanations and to those models where they were able to much quicker assess whether a particular AI prediction is trustworthy and it's actionable than what would be possible if they would not have AI system at all, or they would have an AI system that would simply provide predictions without additional justifications and interpretations. In another example that I'd like to highlight in the rest of my time, it relates to this recent work that we have done on rapid therapeutic innovation. And so this particular project was really motivated by the urgent need of pandemics, where where we are faced with such a public healthcare emergency as pandemic is, there is a tremendous need to develop and deploy safe and effective therapies at unprecedented speed. And because of that, traditional iterative development and experimental clinical testing, approval of new drugs, is often not feasible at all. As I said before, it takes an order of 11 to 16 years to develop a new drug from scratch. So that's not feasible. The challenge is, how can we compress years of work then into months or even weeks through AI, automation, and new data sources? So I will highlight a research project that our team was involved in, and we were leading the AI part of the COVID-19 task force in the US. And during the span of just 3 months, between March 23rd, 2020 and June 15 of the same year, we were able to identify and first implement AI algorithms that our group has been developing, and then put them to use to identify promising drugs that are already approved and are on the market that could be repurposed for COVID-19 patients. And not only identify those drugs, but also engage in rigorous experimental testing to show they're safe and effective. So to this end, the first question that arises whenever you are engaged in an AI project is, what is the data that we'll be using to extract insights that can inform predictions? And so in this particular project, we constructed a very large COVID-19 repurposing knowledge graph that is a data set that consists of information about each and every protein, tens of thousands of proteins in human cells, and how those proteins, those molecules interact with each other in each of the cells. On top of that, we brought in information about every approved drug in the US and the way those drugs work by targeting proteins in human cells and convincing them to take a particular action that could treat the disease. And then we also brought in information about thousands of diseases, common diseases, and how those diseases are treated by approved drugs. And we, together with the FDA, which is the regulatory agency in the US, we looked at that large knowledge graph in the context of known approved indications in telling us what existing drugs are treating what diseases. And given that, our task was, given information about known drug disease treatments, can we identify candidate treatments for COVID-19? Can we produce a ranked list of drugs that are on the market that we could very quickly and efficiently repurpose for COVID-19 patients? And so the first test of the predictions for that particular problem that we did was to evaluate predictions against those drugs that were at that time studied in clinical trials. And so that has already revealed considerable promise of our AI algorithms, and in particular GMeta approach, which is the name of the algorithm used here, relative to other computational methods and non-AI based techniques, shown on the left as a plot of a true positive on an AU-ROG plot, where better performance would be achieved by the algorithm whose line would be closer to the top left corner. And we can see a substantial improvement of GMeta AI approaches over alternative techniques. Kind of that has motivated us to really engage in experimental testing. So our predictions were then fed into experimental systems in National Emerging Infectious Disease Laboratories, which is a level 4 laboratory in Boston that can work with live virus. And what our colleagues there have done, they took top ranked drugs, most promising predictions according to the algorithm, and they have tested them in the lab, first in African monkey cells and then in human cells as well. The result of that, without going in detail, is that by screening these drugs in human cells, results show that we obtained a near 62% success rate, meaning that 62% of drugs that were predicted by the algorithm and tested experimentally were shown to inhibit viral growth. In contrast, we would get only 0.8% success rate if those experimental screening would not be guided by AI algorithms. So this is a concrete example of an order of magnitude better hit rate than alternative approaches. Furthermore, we were very interested in understanding what is defining those set of drugs that were predicted successfully and validated experimentally. So what we found is that 76 out of 77 drugs that were shown to successfully reduce viral infection were drugs that had very specific therapeutic action. The way they exert their medicinal effect was by not binding directly to proteins that the virus attacked. Instead, they were able to find workarounds and essentially target some other proteins in human cells not attacked by the virus, and in turn those proteins interacted and influenced the viral proteins that virus has attacked. And these kind of insights that led to a new class of drugs that we refer to as network drugs would not have been possible without without providing interpretable explanations and justification for why an algorithm has made a particular prediction. So let me conclude by a question and then an answer to that. Is really, I highlighted a few, in particular two vignettes of the work that showed how very concretely, AI algorithms can improve efficiency of drug discovery development research, can lead to new biomedical insights and discoveries that would not be possible otherwise. So the question becomes, how can you here, for example, engage in that research? How can you advance actionable AI for biomedicine problems? So to answer this question, we created an open science initiative called Therapeutics Data Commons. You can visit it by going to the website tdcommons.ai. It's a machine learning foundation for actionable AI. What it allows you to do, it allows you to start working on problems that are real problems in the design, development of safe and effective therapeutics that were identified by domain scientists. And you can design powerful AI ML models for those problems, submit your predictions, and then see how well your model performs in addressing those real open grant challenges in biomedical discovery. And so Therapeutics Data Commons can facilitate algorithmic scientific advance. In therapeutics, it supports the development of novel machine learning theory methods with really a strong bent towards developing foundations and understanding what ML algorithms are most suitable for drug discovery applications, why that would be the case. In just over the span of the last year, we have seen over 55,000 scientists around the world using this resource to practically advance AI and provide AI solutions for real-world problems in discovery of therapeutics. So I will stop here because I'm running already over time. Very happy to take any question if there is any, and you can also learn more about our projects by going to our lab website and also by going to Therapeutics Data Commons. Thank you so much for listening.
Thank you very much, Marinka. For the sake of time and because our next speaker has a commitment after her talk, we're going to have to move on. Thank you for a very informative and actually enlightening presentation, and look for more.